So it gives me great pleasure to introduce Kristen Brennand, Associate Professor, Department of Genetics and Genomic Sciences, who'll give a very brief historical perspective on the topic of cell and gene engineering before introducing the first speaker.
Thank you, Scott. So they asked me to give a historical perspective of research over the last 50 years in cell and gene engineering, and to me it was a really interesting dive. For the students in the room, to think back, what do you think we knew 50 years ago? We knew there were cells, right? And it was early for you guys. We knew there were genes, maybe? Did we know there was DNA? What could we do? And so it's in that framework of just going back and remembering what we knew and what we didn't know 50 years ago. And so in 1968, 50 years ago, we did know that there was DNA, but we didn't know how to move it between cells. And so one of the first discoveries 50 years ago was just that viruses carried DNA and they could transfer it into the cells that you infected, which is the very premise for which gene therapy is actually based on. And then 20 years later, a different type of virus, retroviruses, could also transmit DNA, and that would be stably incorporated. We discovered DNA transfection in 1978. So when you think about gene therapy in patients today, 30, 40 years ago, we couldn't transfect DNA. And today, the Zangi lab is transfecting RNA in vivo into heart tissue. So from 1995 to 2011, virus-based gene therapy clinical trials really finally took hold. So we have therapies for SCID in 1995, for hemophilia in 2011, and one of our keynote speakers, Catherine Hai, was a part of that study, and she'll tell us more, I hope. We have now in 2017 single-dose gene replacement therapy for spinal muscular atrophy, so we are curing a deadly disease before the kids are toddlers. And in the Hajal lab here at Sinai, we have gene therapy in the dish for heart mutations, so here are some cardiomyocytes beating abnormally, and after gene therapy, they're beating normally again. In 1998, we grew the first human embryonic stem cells. So this was Jamie Thompson's lab in Madison. But it was actually a Sinai faculty member, Thomas Iwaka, who in 2003 did the first genetic editing of human embryonic stem cell lines. 2008, so now we're only 10 years ago, is the derivation of the first human-induced pluripotent stem cell lines. The first time we showed that from a patient's skin samples, we could make stem cells from them. Shinya Yamanaka won the Nobel Prize for this just 4 years later, and today I had trouble stopping the list to fit on one page of the number of Sinai labs using human-induced pluripotent stem cells to study disease. 2010, the first embryonic stem cell clinical trial started. And today, so labs all around Sinai are, again, they're making brain neurons and cardiomyocytes to treat brain and heart disease. 2012 to 2013 was when we discovered CRISPR-Cas9. That's just 5 years ago. And so we've discovered it, and now there are labs doing genome-wide CRISPR screening here at Sinai to study viral infection and cancer and more. 2016, the FDA approved the first oligonucleotide drug, Spinraza, for spinal muscular atrophy. And now today, Sinai scientists, together with Semaphore, are preparing for the first in human clinical trial of an oligonucleotide drug therapy targeting VEGF for Alzheimer's. So you see that we're faster and faster, we're piling on discoveries. 2017, universal donor engineered CAR T cells achieve remission of leukemia. And 2018, we're doing it here in our patients. And so that's brought us to the present. And now I'll ask Irini to come up to introduce our first speaker. Thank you.